Hello, everyone. I'm going to be talking about what I would like to do for my PhD. I'm still quite fresh, and I've yet to get my first field season under my belt. But yeah, I'm being supervised by Prof. Alan Ellis, and this project has been generously funded by the Africa Museum and the Dip project. All right, so I'm sure you've heard of these beautiful annual displays of daisies that we get on the west coast and the Namaqualand of South Africa. These come in the springtime after the rainy winter season, and they are truly a spectacle to behold, particularly when we look at them at the community or landscape scale like this. But when we look even more closely, we can see that these daisies have some quite elaborate and detailed spot traits. And this seems to be a pretty unique phenomenon in the daisies globally. And there's mounting evidence that their evolution is driven by the visual systems of their fly pollinators. Okay, so here are just a few of the spotted daisies paired with their main fly pollinators that we find in the Cape, or the Greater Cape Pholistic region. And you might be familiar with stories of this fly here, Megapalpus capensis, in which mate-seeking male flies pollinate this flower here, Diptera diffusa, by being duped into thinking the spots on the florets are females. So the males of this species exhibit a characteristic kind of pinning tactic where whenever they land on the floral spots. And interestingly, this is something that we have observed in other fly taxa as well. So, for example, the males of this Regioglossa and Corsomiza species also hop between flowers with this pinning tactic with a high frequency, and they tend to land on the spots when they do that. So we wonder whether these two Dimorphitheca morphotypes might be exploiting pollinators by similar deceptive means to Diptera diffusa. So when we look at these flies a little more closely, you can notice that the species that exhibit this pinning behavior often lack a sexual dimorphism in their eye structures, right? So here we have two species pairs from the genera Regioglossa and Corsomiza, and notice that it's only the males of Regioglossa edentula and Corsomiza nigripes that have these enlarged eye facets in the dorsal regions of their eyes, whereas Regioglossa nitens and Corsomiza bicolor, the males and the females have very similar looking eyes. So throughout this talk, I've kind of termed the ones with the facet dimorphism FD flies and the ones without the facet dimorphism NFD flies. Okay, so what's the significance of this dimorphism? The literature suggests that these enlarged dorsal facets constitute what is known as a love spot, right? Which is a region of enhanced visual acuity, sensitivity, and motion detection in the fly's visual field. Okay, so this love spot aids in tracking small mobile targets that fly overhead, and so it helps males catch females in flight. But it's also potentially an energetically costly adaptation because it increases the amount of information that has to be processed by the fly's brain, right? So it seems like the transitions between these facet dimorphic and facet non-dimorphic states might be driven by the balance of selection operating through the energy budgets of these flies, right? And there's reason to think that the behavior of the females might be the key to explaining these transitions. Okay, so on the one hand, high definition vision is cool because you are better able to find and catch mates in flight. So for example, one can imagine a scenario where it pays to have the supervision when females are very mobile, so you can use it to catch them in flight, to detect them and catch them, right? But on the other hand, not having HD vision potentially saves you some energy that could be better spent elsewhere. For example, when you're flying between flowers, constantly looking for mates. And we might expect the scenario when females are less mobile and spend most of their time in flowers. So they're in predictable locations. So it makes sense to focus your search patrolling those areas that they frequent. All right? And it may be the case that the daisies exploit this active mate search of NFD flies by evolving these insect-like spot traits that encourage the mate-seeking behavior, forcing flies to check closely enough whether there is actually a potential mate sitting in the flower, right? Whereas potentially this doesn't happen in FD fly species whose eyes are on the skies. Okay, so for my thesis, I will be investigating these ideas, and I have 3 data chapters. The first being an examination of the patterns and the timing of eye evolution in these GCFR fly pollinators. And then I will assess the diversity of mate-seeking behaviors and test their relationships with eye structure. And then finally, I will use visual modeling and behavioral experiments to test the perceptual similarity between flies and floral spots. Okay, and this here is the general design for my thesis. Okay, so we're working the Greater Cape Floristic region here, and we have identified 4 different clades in the GCFR fly pollinator assemblage, that being Regioglossa, Megapulpus, Calynthophora, and Corsomyza. Within each clade, we've got a pair of species, right? One which is facet dimorphic, and one which is not facet dimorphic, and I will be looking at the eye morphology and the behaviors of both sexes throughout. Okay, so let's dive into understanding the patterns of eye evolution in these flies, right? So for this chapter, I have 2 questions. First, have transitions between FD and NFD states evolved repeatedly and consistently? And secondly, what is the timing and tempo of these changes? Okay, so to answer the first question, I will collect flies from my species pairs in the field and subject their heads to micro-CT scanning. Okay, then I will use this 3D ommatidial detection algorithm to isolate the corneal cones, and that will allow me to measure various visual parameters along horizontal and vertical sections in the fly eyes, like this. Right? So the traits that I will measure include the diameter of the facets, the interommatidial angle, which is very important for estimating the acuity of the flies, as well as the field of view, among other traits. Okay, then I will use a multivariate analysis to test for the repeated evolution of or repeated transitions between FD and NFD states, and to test for differences in the effective sex between clades to test that consistency. Okay, so if both hypotheses are supported, we might expect the results to look something like this, right? So all of the open shapes here are FD species, all of the closed shapes are NFD species, females are circles, males are triangles, and generally we would expect, if the pattern is repeated, we would expect males to consistently fall out, separate from the rest of the flies, and in similar magnitude, and that would be the consistency of the pattern. Right? Next, to value the tempo and the timing of eye changes, I will map FD and NFD states across the dated Maribesni phylogeny, which Cass Barker produced for her PhD. And then I will test for the phylogenetic independence of transitions, as well as evaluate the relative support for 4 different evolutionary models to work out the likely timing of these transitions. Okay, but from what we can tell so far, it seems likely that these eye transitions occurred independently in Corsomyza, Megapalpus, and Calynthophora, with an older split in Megapalpus and more recent splits in Corsomyza and Calynthophora. Okay, on to chapter 2, where I will be investigating the mating behaviors of these flies. Here I have 2 questions. Do NFD and FD males exhibit different mate-seeking or courtship behaviors? And secondly, or does the activity of females predict male activity as well? All right. So to answer these questions, I will conduct timed behavioral observations of individual male and female flies in each species pair by just standing and watching and using a dictaphone to describe their behaviors. And the kinds of behaviors that I will describe would be things like the frequency of pinning hops in male flies, aerial chases of moving targets, mating initiation sites, and the proportion of time male and female individuals spend moving between flowers versus feeding, resting, and mating. And I expect to see that FD and NFD male flies exhibit contrasting patterns in terms of pinning visits versus aerial chases. So just as an example of the results, where facet dimorphic species, they tend to give chase more frequently than non-facet dimorphic species, and NFD species tend to exhibit this pinning behavior more frequently. Okay, and then I expect that these differences might also be related to female mobility, such that the amount of time spent in flight by females is negatively related to the amount of time spent in flight by males, with the NFD and FD flies separating clearly along this axis. Okay, and now on to the 3rd chapter, where I will look at the interactions of these flies with floral spots. So here I ask whether NFD and FD flies differ in their interactions with spotted daisies, whether floral spots are perceptually distinguishable from females, and whether males interact in the same way with floral spots as they would with females sitting in flowers. Okay, so to test this, I'll use a data set of behavioral observations collected concurrently with the previous chapter's data set, except here I will focus on the daisy species that these flies visit, whether the species is spotted or not, and the dominant behavior exhibited during each visit. So this could be mating, inspection, feeding, or resting visits. Okay? And here we expect NFD flies to be more strongly associated with spotted daisies than non-spotted ones, and we might also expect NFD male flies to show a much higher frequency of mating and inspection visits than FD males and females. Okay, I think I'm running out of time here, so I'm just going to skip through this. And then, yeah, essentially I would like to also do some binary choice experiments where I test whether flies respond differently to spots in females and flowers using cages, binary choices in cages. For male flies, a choice between 2 different floral manipulations. And there are 3 experiments that I intend to do here. First, testing for preferences between flowers with and without spots, then flowers with the spot removed and replaced with a female fly, and then finally for preferences between spotted flowers and flowers with a spot and a fly in there. And I expect the results to look something like this, where male preferences always favor the presence of either spots or flies, but without discrimination between the two when spotty or fly-bearing flowers are the only choices available. All right, so by investigating the evolution of eye structural diversity, its relationships with mating behavior, and the impacts of mating behavior on flower visitation patterns, I aim to generate an improved understanding of the life histories of key fly pollinators and the processes underlying the weird and interesting floral diversity of South Africa. Thank you very much.
Okay, thank you very much, Arjan. Our screen says time's up, but we'll take one question. Anyone? Okay, no, let's give Chris the credit. Sorry, your conflict of interest, or what is it?
Hey, thanks. I guess, yeah, this is more like a proposal than yeah, so I'm kind of going to treat it like a proposal. So in your explanation about the potential evolutionary consequences of the eye dimorphism and that differentiation between the different ommatidia, you were mentioning something about high resolution versus low resolution, and was it the upper the upper section of the eye that you were saying was going to have higher resolution?
Yes, potentially.
Can you justify that? Because just with regard to the structure of the ommatidia being larger and having fewer of them in the same surface area, that actually would lead to lower resolution. It would lead to higher detection possibility because of higher levels of light entering the ommatidium, but it's not going to lead to more resolution.
Well, so there are several factors at play. Like, I agree with you that, if you have larger facets, that forces the interommatidial angle to be larger, right? And so you can't sample with as high a density, but that effect can be offset by flattening the eye surface, right? And that's something that I showed in this fly here, right? You've got this flattening of the eye surface, which reduces the interommatidial angle, and suddenly you can start to sample in a higher frequency in space, and so potentially you have more acute vision. So really, and that's kind of the use of this micro CT scanning technique, is you can work out all of the geometry and you can figure out whether that section should, in theory, be more acute than below.